Hello everyone, and welcome to today's webinar, Leveraging Acoustic and Tip-Based Liquid Handling to Increase SARS-CoV-2 Sequencing Throughput. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Beckman Coulter Life Sciences. To learn more, visit beckman.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Eric Chow, Assistant Professor, University of California, San Francisco. Dr. Chow, you may now begin your presentation.
Thank you for the opportunity to speak today. Again, my name is Eric Chow, and I'm with the Center for Advanced Technology in the Biochemistry and Biophysics Department at the University of California, San Francisco. And today I'm going to talk to you about work that we've done to leverage acoustic and tip-based liquid handling to increase our SARS-CoV-2 sequencing throughput. First slide is just a quick disclaimer. You can take a look at this. And again, the focus of today's talk is on COVID sequencing and the role it plays in COVID genomic surveillance and how we can scale this up, just because there's an important need. On the left-hand side is a graph just showing you the number of cases that have been, present in the United Kingdom, one of the countries with the kind of best COVID surveillance efforts going on. And you can see that while there's a huge increase at the beginning of the year, there's a rapid drop, and things were looking pretty good until recent weeks where you started to see a spike in infections. And a lot of this is really driven by the appearance of this Delta variant first identified in India. And you can see this on the panel on the right-hand side, where you can take a look at the fraction of cases that are made up of different strains. So initially, the Alpha variant in blue made up most of the infections in the UK, but you can see just a few months ago, the Delta variant in yellow started to show up and is now making the majority of the cases that are coming up and being detected. And again, it's thought that this Delta variant may be 50% more infective than the Alpha variant, which was, again, itself 50% more infective than the original strain. And so this demonstrates the importance of being able to do genomic surveillance to see what type of strains are circulating out there and which ones may be increasing frequency, which may be an indicator of increased transmissibility. So it's estimated that to do the minimum amount of sequencing required to perform genomic surveillance is 5% of positive samples need to be sequenced to be able to track these variants. If you can sequence a higher amount, greater than 5%, you'd be able to detect new variants that are spreading faster and earlier. It's also important to sequence positive samples from vaccinated individuals to identify potential variants that could escape the vaccine. Just a quick note, all the major vaccines in use are still very effective against all identified variants, but there's always a worry that a variant may come about that can escape vaccine protection. And genomic surveillance is going to be important to identify those potential variants. So worldwide surveillance is going to require tens of thousands of samples per day if you assume that you just want to sequence 5% of all cases. So this works out to about 20,000 samples per day. Again, this is really the minimum that you would want to sequence. Ideally, you'd be able to sequence a greater fraction of cases and be able to identify variants that are arising sooner. Again, there's also a need to sequence additional samples from infections of individuals who've been vaccinated to identify potential escape variants. And we need a mix of both really high throughput centralized sequencing centers and lower throughput decentralized methods to be able to cover all the different areas that require genomic surveillance. There are currently several main methods of SARS-CoV-2 sequencing. There are many, many methods being developed all the time, but I'm just listing probably the three major ones that are being used. Originally, metagenomic next generation sequencing was the first one that was used. Essentially, this just takes a sample and sequences everything in there and asks what's present. And so this was a method that had been used to sequence and identify different types of pathogens present. And until SARS-CoV-2 specific methods came about, this was the main method people were using to sequence sequencing samples. There are some challenges in that it's not quite as sensitive for high CT samples or low titer samples. It can also be expensive and somewhat laborious. What came out more recently was something from the Arctic Network, a group of labs that have published a lot of great and amazing protocols, not just for SARS-CoV-2, but for a lot of other pathogens in the previous few years. And with their scheme, they actually use a multiplex PCR reaction to amplify different amplicons that tile the entire pathogen. So what they typically do is split the amplicons into the even and odd numbers. And then all the even number amplicons are in one pool, all the odd number amplicons are another pool. After you do this amplification, it goes into a traditional library preparation DAD adapters and barcodes. And then you can put this onto a sequencer. And this usually takes a bit more time in preparation, mainly due to the length of time for the multiplex PCR reaction, and can also be a little laborious as well, just because of the downstream library prep. A new method that came out probably about last summer, was when the preprint came out, was the tailed Arctic method. And so this is very similar to the Arctic method, but instead of using standard primers for the multiplex PCR, they added adapter tails onto the end of the primers. And this allows you to essentially perform your library prep with two sequential PCRs. The first PCR will amplify your SARS-CoV-2 targets and introduce Illumina read 1 or read 2 tails, or for other sequencers. And then the second PCR that's performed will finish the library prep process, adding in the rest of the adapters, as well as indexing barcodes so you can pull samples together. The one downside of this method, while it's fast and a lot easier on the preparation time, it required a longer sequencing method, so paired in 250 sequencing on Illumina platforms, which takes a bit more time than the traditional paired in 150 sequencing that had been used. And it also requires 4 separate multiplex PCRs to amplify the SARS-CoV-2 genome. And I'll talk about that in a couple slides. But due to speed and cost, most groups are currently using one of the Arctic methods now. And again, both of them have limitations. And so what we really want in a DL method is something that's really high throughput, has rapid turnaround time, reduced tip usage, as I think most people on this this seminar today realize that there are supply chain issues with tips. And ideally, it's deployable on all Illumina sequencers, which means that the method has to be able to run on paired in 150 sequencing. The paired in 250 sequencing used in the current tailed Arctic method requires paired in 250 sequencing, which is restricted to the MiSeq and the low throughput NovaSeq kits. Ideally, the method is going to be low cost as well. And so in our minds, the ideal method would be based on the tailed Arctic method, just because the workflow is really simple and really high throughput, but would only require paired in 150 sequencing. And I'm going to talk a little bit more about the kind of development of the tailed Arctic method. And this has been a really great method to really increase throughput, just because sample preparation was really a big bottleneck. And so on the top panel in blue, what I'm showing you is a list of amplicon coverage of the original Arctic method that does multiplex amplification in 2 pools, and then sample prep afterwards. And you can see it's relatively even coverage across the amplicons. You do have some dropouts. In the first version of the tailed Arctic method, they use 2 different pools, and the handles that they attached onto their primers are Nextera read 1, read 2 primer handles. And you can see the coverage looked a little bit more uneven. You can see it's a bit spikier compared to the standard Arctic method. And so what the authors did was they, instead of using 2 pools, they split the amplicons into 4 pools and were able to get a bit more even coverage. So the downside of this is that instead of 2 multiplex PCRs per sample, you have to do 4 multiplex PCRs per sample and then combine and pool them. And so in the CAT, what we decided to do was tried doing one thing. First thing is that we realized that the Nextera tails are very long, and Nextera primers also have 19 bases homology in the underlying section between the read 1 and read 2 sequences. And so we realized this could probably cause problems with the multiplex PCR. And so what we decided to do was swap these out for TruSeq read 1 and read 2 tails that were shortened. So they're only 17 and 18 bases in length versus 33 and 34. And the region of homology between the read 1 and read 2 primers is quite a bit shorter for TruSeq compared to Nextera. And to rapidly prototype these, we actually ordered O-pools from IDT. So we didn't have to order a large set of individual oligos. We could order a small set and have a relatively quick turnaround time to begin testing this process. And so results on the bottom is just from one kind of representative sample that we were able to get fairly even coverage. And this was with only 2 pools instead of 4 multiplex PCR pools for each sample. And so we see characteristic dropouts or lower coverage in these areas that are seen with the traditional Arctic method. And so this looked relatively good. So we decided to move forward with 384 samples that had been processed using the traditional Arctic method. And what I'm showing you here is our results. So this is a plot of average viral CT versus percent of the genome called. And for our work, we've been using a 92% cutoff of genome coverage to delineate passing versus failed samples. And so the passing samples are in blue, and the failed samples that didn't hit that 92% coverage are in orange. And so you can see that there's definitely a strong CT dependence here. And so we got about 72% of samples passing, which is a little bit lower than the standard Arctic method that these samples were run on as well. And so our summary of this method was that, the tailed 400-based method using these TruSeq tails seemed to work. We would like to have kind of improved sample passing rates, but there's still a downside in that it required paired end sequencing, which requires about 2 days on the NovaSeq SP flow cell. So at the time we were working on this new tailed method, we saw that Josh Quick's group also had a 275 base pair Arctic primer set that showed even more even coverage than the 400 base pair set. But with their protocol, they went through the traditional library prep after the multiplex PCR. And so we decided to do, since our TruSeq tailed versions seem to work better than the Nextera tailed versions, we ordered again an O-pool using a tailed version of these 275 base pair primers with our TruSeq sequences. And we ordered these in two modes. First was as an O-pool so that we can begin testing as soon as possible because of the fast turnaround time. And we also ordered each of the primers individually in plates just so that we could scale up to about 100,000 reactions total. We didn't want to have to revalidate primers, several times. So we did put in a big order with IDT. And these can actually be delivered into echo plates, which is an acoustic liquid handler I'll tell you in a moment. And there are certain advantages to ordering in an echo plate. Namely, you can create pools almost on the fly and change your ratios of different primers, which can be important to normalize coverage with these multiplex PCRs. And so the tailed 275 base method actually gave more even coverage than the tailed 400 base method, which was also seen in Josh Quick's group using the standard Arctic procedures of library prep, comparing the 400 and 275 base protocol. So these are 2 samples. Top one was run using the tailed 400 base pair protocol I showed earlier, and the bottom is the tailed 275 base pair method that we worked on. And so this looked really promising. So we ran a couple more samples just to do some comparisons that were run using the standard Arctic method of multiplex amplification and then library prep versus our tailed 400 base pair prep versus the tailed 275 base prep. And you can see that while the standard Arctic performed better than our tailed 400 base method, the tailed 275 base method actually worked a lot better for these representative samples, and that most of them gave greater than 92% coverage of the SARS CoV-2 genome. And again, this is depicted in these green boxes. The one sample that didn't pass actually had some evaporation issues with the plate, so we're not sure if this is a true failure or not. But it looked really promising. We decided to move forward with this method. And so to really increase throughput, we wanted to really automate this method to be able to sequence as many samples as possible in a high throughput manner. And so that really requires the use of automation. And there are several advantages to automation besides increased throughput. These include increased reproducibility, prevents user error that can mix up samples, and also preserves staff sanity. You can imagine trying to work in 96 or 384 well plates with multi-channel pipe headers, and it's just going to be a huge headache and a lot of opportunities for mistakes. The other thing we wanted to do was perform our reactions in 384 well format, again for high throughput production. And so this required the need to move samples from 96 to 384 well format. We also wanted to minimize tip usage due to supply chain issues. And lastly, we wanted to make sure that we minimize contamination. So a lot of people on this call may have worked in, just other genomics projects in the past. But when you're dealing with pathogen sequencing, and SARS CoV-2 sequencing, it's very different from other standard NGS experiments where you might be working with blood samples or RNA-seq input samples. And for a lot of those samples, you probably have relatively equal inputs across all your samples. So if you have a little bit of cross-contamination between samples, it's just something you don't see routinely. It's difficult to detect. But with SARS-CoV-2, samples that are positive can vary in CTs by up to 30 cycles, is what we've seen. And so this represents about a billion-fold difference in abundance from the most concentrated sample to the least concentrated sample. And so you have to be very, very careful about contamination. And we feel that using some of the steps with the acoustic liquid handler, we actually minimize chances of contamination. I'll talk about that in a couple of slides. So in this next slide is the automation workflow that we developed using various liquid handlers. And so I'm going to walk you through this process, and it also gives you an overview of the protocol itself as well. And there'll be links to the detailed protocol in the resources section of this webinar that you can access after this call. So first we start off with RNA, and so this can either be in 384 well plates or 96 well plates. And in our case, our samples are in 96 well plates. So we have a Biomech FX with a 96 well head and 8-channel span head that we use to essentially consolidate 4 96 well plates into a single 384 well plate with our reverse transcription master mix. And this is actually done in a Echo Acoustic low dead volume source plate, and I'll talk about that in the next 2 slides. And so reverse transcription occurs in this Echo source plate, and once that's done, this source plate goes into the Echo 525 acoustic liquid handler, and this system uses sound to transfer small volumes, 25 nanoliter droplets of liquid, from a source plate into a destination plate. And our destination plates here are our 275 base tailed multiplex PCR plates. So in one plate, we have all the odd amplicons. In one other plate, we have all of the even amplicons. And so we transfer 1 microliter of the cDNA product into each of these plates. These are then thermocycled to amplify up the SARS-CoV-2 targets. And then we use a Biomech i7 with a 384 well head and a span 8 head to combine and dilute the amplified cDNA products and to stamp this into an indexing PCR plate. After this indexing PCR plate is thermocycled, it then is transferred using the Biomech i7 into another Echo low dead volume plate. And we use the Echo to pool an equal volume of each sample. We pool 500 nanoliters of each sample into a new destination plate, and then combine that into a single tube where we do a single bead-based cleanup. So after this bead-based cleanup, the samples are quantified and QC'd and go onto the sequencer. And so just want to note that in this process, we're actually only using 2 tips total for this procedure. The first tip is used to transfer RNA from the 96 well plate into the low dead volume plate. And the second tip is used to combine the amplicons, dilute them, and spot them into the indexing PCR. And the same box of tips is then used to transfer the amplified bar-coded samples into the Echo LDV plate for transfer. And minimizing this tip usage really helped with our throughput as well. And we haven't had any issues with tip supplies. So in the next slide, I just wanted to briefly go over how the Echo Acoustic Liquid Handler works, because a lot of people may not be familiar with it. It's essentially a random access liquid handler that transfers liquids using sound. And so the way this works is you have a source plate that's an acoustic qualified plate, and there are a couple different formats in 3D 4-well format, and also a 6-well reservoir for the Echo 525 that we're using. There are also higher systems to transfer smaller volumes that can use 1536-well source plates. But for us, the Echo 525 works very well. It's kind of a nice volume regime. So this source plate will go into the Echo, and then next you put on your destination plate. And in our case, we've been using these 384-well PCR plates. This is put on the this little platform. The platform will grab and grip the plate, and then it'll flip and rotate 180 degrees so that this plate now faces down. This source plate will retract into the instrument, and in the instrument, the source plate down on the bottom and the destination plate that's flipped upside down facing it are in really close proximity. And below the source plate is a transducer that will send pings of sound energy into the source well, and this causes really tiny 25 nanoliter droplets to get ejected from the meniscus straight up. And because the source plate and destination plates are so close facing each other, there's very little chance of cross-contamination happening, because a 25 nanoliter droplet essentially flies straight up, exits the source well, and enters the destination well. And so the system is really quick. It can transfer several hundred droplets per second. So copying a 3D 4-well plate and stamping out 1 microliter only takes a few minutes, and again, doesn't use any tips. We've had some questions about how we perform our reverse transcription incubation in the Echo low dead volume plate. And so optimally you would have a flat block thermocycler, but there aren't many of those around typically in labs. And so what we did was we used a Birad C1000 384-well thermocycler that we had, and we essentially modified it by buying some things off of Amazon, essentially, because it was the fastest way to get supplies. And so first thing we did was we took a thin thermal pad. And so this is a relatively soft, thermally conductive pad that we cut to size to put on top of the 384-well thermocycler. And then on top of that, we put a thin aluminum shim stock on top to give it a nice flat surface. And this is how we modified our C1000 thermocycler. And then we put an Echo LDV plate on top. That's a little hard to see in the right-hand side, but it's a clear plate that's now sitting on top of the shim that's sitting on top of the thermal pad. So the thermal pad will help with heat transfer, and the shim provides a nice flat surface for the Echo LDV plate to sit on top of. And we compared both running RT reactions in PCR plates as well as the Echo LDV plates, and we didn't see any changes in performance. And again, the benefit here is using one less piece of labware and less tips that are required to do your downstream transfer. And so in this next slide, what I'm showing you on the left-hand side is the results I showed earlier from our tailed 400 base method, and on the right-hand side are those same samples that have been run using our tailed 275 base method. And so if you recall, with the tailed 400 base method, we only had a 72% success rate, but with the tailed 275 method, we actually had a 90% success rate. Again, this is dictated by a number of samples that have greater than 92% coverage of the SARS-CoV-2 genome. And when we actually look at samples below CT of 30, our success rate is actually 97.3% with this test set. And down the bottom is a little chart where we kind of broke down the samples according to CT bins. And you can see at the lower CTs, you were getting very high recovery of samples. And it's really once you get to CTs above 30 that the success rate starts to drop a bit. But we've been very happy with this protocol and been using it to run a lot more samples. And so with the current status is we're currently running 768 samples twice a week. So our batches are 2 3D 4-well plates of samples at a time. And we've been seeing above a 95% success rate, again, determined by more than 92% coverage. And so our biggest limitation right now is actually not in our liquid handling, but it's more in staff time. So the staff have to perform other duties as well. And so they can only handle so many runs, per week because of their other duties. But the liquid handlers, by our calculations, if you had enough staff, you could easily process 384 plates of samples per day. We have pretty rapid turnaround time with this method. The 384 or 768 samples can be prepped and ready for sequencing in about 8 hours. So it's about one shift or one workday. And again, really importantly, we have reduced tip usage. We started off with 3 tips per sample, but realized we could actually get that down to 2 tips per sample. And the method is fairly low cost. It uses off-the-shelf reagents. And because we're working in 3D4-well format, we're working with 10 microliter reactions. And so we have 1 RT reaction and 3 PCR reactions. So the 2 multiplex reactions and 1 indexing PCR reaction. And again, because we have the echo, it makes pooling samples really easy, which means that we can pool our amplified samples before cleanup and do a single bead-based cleanup on a combination of all 384 samples. And so a lot of this has been really enabled by combining the unique aspects of our various Beckman liquid handlers. A big one is the Echo 525. It allows us to pull samples really quickly. It also allows us to transfer small volumes with good accuracy, as well as minimizing contamination. And then we've been able to use our Biomech i7 and FXs to handle our 96 and 384 well transfers. Sometimes I get the question that if I was going to start over, how would I set this up? And if that was the case, I would probably just start off with the Echo 525 and then a single Biomech i7 with a dual arm that can be configured with both a 3D4 well head and a 96 well head, because that would handle all of our kind of liquid handling needs. So instead of having 3 instruments, we could actually make do with 2 instruments. All right, so this has been great, and so I've been thinking about some other ways of increasing throughput. Mainly, how do you deal with maybe the being shorthanded in terms of staff time, and how to really leverage the amount of capacity on the automation platforms. And so one piece of automation that I have in another lab that's not the CAT is a Beckman Access system, and so this has an Echo 525 liquid handler attached to it, as well as an Access robotic arm that can feed many plates, up to 80 or 100 plates, depending on your configuration, stacked in hotels. The system also has an automated sealer and peeler and a plate centrifuge, as well as a bulk dispenser that can be used to transfer small volumes of bulk liquid. And so one thing that I envision happening is if we want to really increase throughput while dealing with staff limitations, is to use a platform like the Beckman Access system that can handle preparing your master mixes and dispensing these into many 384 plates. These could be your Arctic PCR amplicons 1 or 2, or your indexing PCR, or your RT plate. And then you can have samples in either 384 well Echo plates or on your tip-based liquid handler to transfer them in. But the system could, on the fly, create your mixes for you. And so, again, all of our amplifications are using a standard PCR mix, and we just have a different type of primer mix in there. And so those could be located in acoustic source wells and source plates that are automatically fed into the liquid handler to create whatever plates you need kind of on demand, and also be able to handle many plates at a time. Instead of the 1 or 2 3D4 plates that we're running in our current batches. And so this has the ability to really increase throughput to that next scale. So we're interested in playing around with ideas like this. And so with that, I'd like to acknowledge a lot of the staff that did the work. This was done in the UCSF Center for Advanced Technology, and Delcy Martinez and Tyler Miyazaki were really instrumental in doing the work and getting this done. I'd like to thank Joe DeRisi and his lab. They provided the initial 384 samples that we compared, as well as the data using the standard Arctic method. Amy Kissler from CDC BioHub for great conversations. I'd like to really thank the IDSeq team from the Chan Zuckerberg Initiative, that developed essentially a cloud-based method that's free for anybody to use to do SARS CoV-2 genome analysis. And so this is how we've been calculating our coverage, and they've been great in terms of uploading our protocol, our specific protocol, into their analysis platform. And lastly, I'd also like to thank all the members of the Arctic network. The work that we're doing here with this method is really based on all the groundwork that they've been doing, again, not just for SARS CoV-2, but even before SARS CoV-2 with things like Zika and dengue. And so they've provided a really great framework to do a lot of this high throughput sequencing that most people around the world are now using for SARS CoV-2. And with that, thank you for listening. I'm happy to take any questions.
Thank you, Eric, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Okay, let's get started. Our first question is, you mentioned improved throughput using this method. In what way did ECHO help you with improving your throughput?
Yeah, that's a great question. So I'd say there are a couple areas where the ECHO acoustic liquid handler really helped with throughput. First of all, again, I mentioned, I think a couple times that it really decreases tip usage, which can, be a limiting factor right now for, I know a lot of groups are running into tip shortages, which is preventing work. And without tips, you've got no throughput at all. And so by leveraging the ECHO, we've actually been able to cut down our tip usage per sample to just 2 tips for each. And by decreasing the amount of tips that we need, we haven't run into any supply constraints. The second area that it really helps with throughput is, I'd say, in the pooling step. So at some point, we need to pool all 3D4-well samples together. And so that can either use up a lot of tips as well as take up a lot of time if you're using the traditional tip-based liquid handler. But with the ECHO, that pooling only takes about 2 minutes. And so this has really helped as well. And we also feel like the ECHO, by the nature of how it does the liquid transfers, where the source and destination plates are really close together, you really minimize the chance of contamination. And often if you have contamination in your controls, what we and most other groups will do is have to repeat the run. And so by minimizing contamination, you minimize the number of reruns that you have to do, which also greatly increases throughput. The last thing we haven't done yet that we're looking to do is how to decrease our volumes. Our current reaction volumes are currently 10 microliters, and that's kind of a nice place to work with tip-based liquid handlers. But again, since we're using the ECHO, we think there's a possibility of decreasing that by several fold. And that's probably next on our list to help increase our throughput.
Thank you, Eric. Our next question, how much volume can you hold in a destination plate upside down when pooling, and what type of plate are you using as the destination plate?
Yeah, so for the work that we do, we are using BIRAD 96 or 3D4-well plates for our destination plates. And really with the ECHO, you can use pretty much any type of SBS format plate that you want to work with, as long as I think they're below like 14 millimeters in height. That's the only limitation. But pretty much any fully skirted PCR plate is going to work, and we go up to 15 microliters in volume for the destination plate, just so we don't fill it up too much. And we just want to make sure that you don't fill it up so much that you get dripping at the end. So with 15 microliters is a very safe volume that we haven't seen any drips or anything like that happening with the system.
Thank you, Eric. Our next question, after the pandemic, how do you see using this workflow to sequence other pathogens?
Yeah, so this is something we've been thinking about as well. So the CAT's done a lot of kind of metagenomic sequencing in the past. We'll try to look for other pathogens, and SARS CoV-2 was our first experience with this Arctic network protocol and method. And something we realized is that what's great about this method is that it's really a framework. You know, you can use essentially the same procedures for any pathogen. You just design a new set of multiplex primers. Your reaction mixes are all identical, you know, for all the different steps, right? Whether it's an RT reaction, a multiplex PCR reaction, or an indexing PCR reaction. So the only difference is your samples and then your primer sets. And so once you've designed your primer sets, within the same run, again, leveraging the kind of random access nature of the echo, you could use the echo to process many different types of samples on a single plate, because you can use the echo to spot out your multiplex PCR mix into the wells that you need for your particular pathogen. And again, for those who haven't worked with an echo or a liquid handler with like a span 8 multi-channel head that can do cherry picking, what you do is you just feed a spreadsheet that just instructs the system what type of sample is in what well. And that'll tell the system which primer set to deposit into that well. And so we think that could be, you know, one way of setting this up, especially if you have a higher throughput, you know, access system where you can load multiple plates in hotels at once. Plates contain your samples, your destination RT plates or PCR plates, as well as source plates containing your indexing primers and your multiplex PCR primers for different pathogens.
All right, thank you, Eric. Our next question. You mentioned you only use 2 tips. How many in total if you also count the extraction part?
Yeah, that's a good question. So that's going to depend on your method of extraction. And so, again, this protocol that I'm describing using 2 tips is only starting from purified RNA. So we haven't tried doing any type of direct amplification from, say, like saliva or heat inactivated extracts. But I've seen different protocols for RNA extraction ranging from as many as, you know, 14, 15 tips. All the way down to 2 tips, depending on the protocol that you're using. But you know, what we think would be a workflow would be most groups would do qPCR testing that would require RNA extraction anyways, and you have RNA left over. And after you get your positive hits determined by qPCR, you can go back to the source plates and pull out the purified RNA, and then go into your sequencing workflow.
All right, thank you, Eric. Our next question. Can you talk a little bit of the liquid handler software? How was your experience programming the liquid handlers?
Yeah, so you know, one thing we really like about the Beckman systems is the kind of ease of use. So the CAD is actually an open access technology facility. We typically don't do a sample prep, but you know, I think the pandemic kind of changed the roles we all play, and so we kind of jumped in to do SARS CoV-2 sequencing because of the need. But traditionally in the CAT, all the instruments that we have, including our liquid handlers, are used by anybody on campus, and we provide training. And so for the tip-based liquid handlers on the FX and the I series, we typically just do a 1-hour training with users who want to learn to use the platform. And that's enough to get them going, to start writing their own protocols. So it's relatively straightforward because of the kind of graphical user interface. You don't have to know much or anything about scripting, for instance, or programming. On the echoes, even easier. It's a little less dangerous because you don't have to worry about tip crashes, right? Because you're not working with tips. And then our training for that ranges from 30 minutes to 1 hour to get people started. So it's pretty easy to get started on the protocol. And start to program these instruments, which we really like in the context of a core facility with many users.
All right, thank you again, Eric. Next question. What volume did you run your RT reaction?
Yeah, so all of our reaction volumes were 10 microliters. So these included the RT reaction that was run in the Echo low dead volume plate. So we started out with 2 microliters of the 5X. It was this LunaScript master mix that we use, and then we add 8 microliters of RNA on top of that. So that was used in that first Biomech FX 96 well liquid handler to do that transfer. And then the subsequent multiplex PCRs were 10 microliters as well as the indexing PCR. And again, I think there's definitely a lot of room to further decrease those volumes if you want to drop costs, just because of the ease of lower volume transfers with the Echo. And so that's something we're playing around with as well.
Great. Thank you again, Eric. We have time for one more question. Do you anticipate decrease of your sequencing throughput as the pandemic winds down in North America?
Yeah, that would be the hope, right? That we can all get back to our normal lives. But I would say that even if case rates go down by a lot, right now I don't even know if in this country we're sequencing 5% of all the positives. So ideally you would sequence as many of the positive cases as you can. So I'd say that even if you were to decrease the number of positive cases by another 20-fold, we might be able to sequence 100% of all the samples. So I'd say that I don't think anytime soon we'll be kind of decreasing the need for sequencing. Again, I think it's going to be important to also continue sequencing positive cases, especially among vaccinated individuals. And to be honest, we totally expect to see positive cases among vaccinated individuals just because the vaccines aren't 100% perfect. So what we want to do is just make sure that there isn't some maybe nasty variant that can escape the vaccine. Again, there's no variant that escapes the vaccine, so everyone should still go out and get vaccinated. But I think the surveillance is going to be important just to detect if that does happen. So I'd say that we're going to continue sequencing for the time going forward, and there's also a need for doing a lot of sequencing outside of North America as well, just around the world. So hopefully some of these methods and platforms can become a use for people outside of North America too.
All right, thanks again, Eric. Do you have any final comments for our audience before we go?
No, I just wanted to thank the audience for tuning in today and listening, and then also just wanted to again acknowledge the people who worked on this project, and also again thank the groups around working around the Arctic network. So there's a group of individuals who've been working for several years on other pathogens and really shifted focus on SARS CoV-2. And have really built a lot of great protocols and resources and shared them widely just to make it accessible to all groups. So I definitely want to thank them for all their hard work as well.
Well, thank you again, Dr. Eric Chow, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.